IB Bio Photosynthesis Part IV is the fourth of 4 movies that cover photosynthesis. Part 4 focuses on the light-independent reactions. This movie covers higher-level material only. The essential ideas are light energy is converted into chemical energy, and secondly, photosynthesis uses the energy in sunlight to produce the chemical energy needed for life. Here is an outline of the movies available for photosynthesis. Only the first 2 are mandatory for SL students. Use this outline to find the movie you need for the review of material. This movie is focused here. The light-dependent reactions were the focus of Part 3. Red and blue wavelengths are absorbed by chlorophyll to bump electrons to higher orbitals. Water is photolysed. Photosystems 2 and 1 and the electron transport systems produce ATP and NADPH. ATP and NADPH provide the energy and reducing power to fix carbon dioxide in the light-independent reactions, which is the focus of this movie. ATP and NADPH from the light-dependent reactions provide the energy and the reducing power to fix carbon dioxide into carbohydrates in the light-independent reactions, also known as the Calvin cycle. While the light-dependent reactions take place on the thylakoid membranes, the light-independent reactions occur in the stroma of the chloroplast. Carbon dioxide is fixed into carbohydrates through a series of reactions involving a molecule known as RUBP, ribulose bisphosphate. RUBP accepts carbon dioxide in a carboxylation reaction. Stay tuned. Here is the detailed IB syllabus statement that drives nearly all of this movie. Explain the light-independent reactions, the Calvin cycle, including the roles of ribulose bisphosphate, RUBP, RUBP carboxylase, also known as Rubisco, the reduction of glycerate 3-phosphate to triose phosphate, NADPH, ATP, the regeneration of RUBP, and the synthesis of carbohydrate. I will present the Calvin cycle in the most simple terms possible. Pay close attention to the language I use and the language provided on the slides. The light-independent reactions, also known as the Calvin cycle, is a cycle because RUBP, ribulose bisphosphate, is joined to carbon dioxide to form glycerate 3-phosphate, but RUBP is reformed, regenerated by the end of the cycle. I tend to teach the Calvin cycle starting with the uptake of 3 molecules of carbon dioxide because the very first carbohydrate formed is a 3-carbon molecule, also called triose phosphate. It takes 3 carbon dioxide molecules to form triose phosphate. It's a 3-carbon carbohydrate. RUBP is carboxylated by the enzyme Rubisco to form glycerate 3-phosphate. This is the first primary product. Rubisco is a critical enzyme in photosynthesis and is the most abundant protein on Earth. Glycerate 3-phosphate is reduced to triose phosphate through a series of steps involving intermediates. ATP provides energy, and NADPH provides reducing power, as you can see in these 2 steps. Glycerate 3-phosphate is energized by ATP and reduced by NADPH to triose phosphate. Triose phosphate is the first sugar product. It's a 3-carbon carbohydrate, and it is easily converted to 6-carbon glucose. Glucose is the monomer of starch. Now let's take a look at what's happened here. 3 carbons have come into the cycle, and 3 carbons in the form of triose phosphate have been pulled out of the cycle. Triose phosphate is a 3-carbon carbohydrate, but it is easily synthesized to form a 6-carbon glucose. From here, all that's left to complete the cycle is to reform RUBP. The regeneration of RUBP is required for the cycle to continue, and triose phosphate molecules are used to regenerate RUBP using ATP as an energy source. Another approach toward understanding the Calvin cycle would be to count carbons. RUBP is a 5-carbon molecule, and 3 RUBPs would be required to join with 3 carbon dioxide molecules. So 15 carbons start the cycle, 3 carbons are added. So now the cycle has 18 total carbons. After the RUBP joins carbon dioxide, there's some molecular rearrangement, forming 6 3-carbon glycerate 3-phosphate molecules. But still, we have 18 carbons total. ATP and NADPH reduce glycerate 3-phosphate to triose phosphate, but there are still 18 carbons in the cycle. Now, 1 3-carbon triose phosphate is removed, leaving 15 carbons in the cycle. Some rearrangement of the 5 triose phosphate molecules results in 3 5-carbon RUBP molecules, 15 carbons ready to start the cycle again. In case you wanted to see the molecules more intimately and you're attracted to the carbon counting concept, take a close look at this slide. Here is RUBP, and you can see that it is a 5-carbon molecule. Add 1 carbon dioxide, now 6 carbons total in the system, and along with some rearrangement, you get 2 molecules of the 3-carbon glycerate 3-phosphate. Some ATP gets you to an intermediate, and the reducing power of NADPH gets you to the product of triose phosphate, a 3-carbon molecule. Triose phosphate is used to produce carbohydrates, and triose phosphate, along with some energy, ATP, is used to regenerate RUBP. Now keep in mind, if you look up the Calvin cycle in an effort to get to know it better, you might find some of the terms to be different. Be patient with this. The terms that I've used in the previous slides are those that the IB expects you to know. But glycerate 3-phosphate is also known as 3-phosphoglycerate, and TP is also known as glyceraldehyde 3-phosphate, aka G3P. G3P, also known as triose phosphate, is the first carbohydrate formed in the Calvin cycle. Here is a diagram of the Calvin cycle produced by an IB study guide. If you prefer the simplicity of the black line drawing, spend some time studying this diagram. The various fates of the triose phosphate molecule are 1. To regenerate RUBP, 2. To synthesize glucose, and 3. Potentially enter respiration in glycolysis. What, you might say? Stay tuned. You should recognize this diagram as glycolysis. This intermediate, the intermediate shortly after the lysis step in glycolysis, is triose phosphate, and this intermediate is glycerate 3-phosphate. One of the fates of triose phosphate from the Calvin cycle, other than regenerating RUBP or being synthesized into large organic molecules like glucose, would be to enter glycolysis in cell respiration processes right here. Glycolysis in the cytoplasm, you should remember, has the very same intermediates as the Calvin cycle. Isn't that interesting? So again, if you want to study the Calvin cycle in more detail, here is a professionally made slide. As I mentioned earlier, you will find some differences in terminology. Study this slide to reinforce your understanding of the Calvin cycle. Here is another professionally produced Calvin cycle diagram for you to study. Stop the movie now if you wish to dig into the detail of the molecules involved. So let's conduct a summary of the atoms involved in photosynthesis. Carbon from carbon dioxide is used to make glucose. Some of the oxygen in carbon dioxide is also used to make glucose. Hydrogen from water is used to synthesize glucose, but some of the hydrogen also remains in water. All of the oxygen released as a waste product from photosynthesis comes from the photolysis of water. Hydrogen ions and electrons carried by NADPH, along with ATP and carbon dioxide, are required to reduce carbon dioxide to glucose. As I've already mentioned, glycolysis, as the first stage of respiration, has the very same intermediates as the Calvin cycle. And remember that glycolysis occurs in the cytoplasm. The first intermediate after the lysis of the fructose 1,6-bisphosphate is triose phosphate. And this intermediate is glycerate 3-phosphate. This is the first significant molecule after RUBP joins with carbon dioxide. The organic intermediates of both the Calvin cycle and glycolysis are central to the metabolism of many biomolecules, glycerol to lipids, fatty acids to lipids, and amino acids to proteins. So let's transition to a new IB syllabus statement. Explain Calvin's experiment to elucidate the carboxylation of RUBP. Calvin received the Nobel Prize in 1961 for his well-known lollipop experiment, in which he was able to elucidate the stable intermediate carbon molecules in photosynthesis. Calvin, seen here on the right, with his equipment on the left, used new techniques in his work to elucidate the stable intermediate carbon molecules in the Calvin cycle. The new techniques were radioactive labeling of carbon, two-dimensional chromatography, and autoradiography. Let's take a look. Calvin set up a closed system with photosynthetic algae that he added here. He provided water, provided carbon dioxide, some radioactive carbon-14, and, of course, light. At varying time intervals, on the order of seconds, he would rapidly terminate the reaction by withdrawing algae into hot methanol, immediately killing the samples and thus preserving whatever products had been formed. The heat-killed samples were then analyzed for the presence of organic intermediates that had incorporated the radioactive carbon-14. Then Calvin separated all of the molecules formed using two-dimensional chromatography. He separated the molecules in one direction with a single spot here, solvent running up the paper, producing a set of molecules or spots, smudges on the paper as so. Then he turned the paper 90 degrees, used a different solvent, and separated these spots from each other. Separating the molecules in two directions, as you've seen here, provides greater resolution to the molecules formed as intermediates in the Calvin cycle. So Calvin would then expose the chromatography paper to a sensitive film that would display the presence of radioactive carbon molecules. This was known as autoradiography. The radioactive spots on the film were identified, and those images can be seen in this slide. Remember, he ran the photosynthetic reactions for varying periods of time, stopping the reactions using the hot methanol at specific time periods as a mechanism to determine the sequence of molecules formed. After 5 seconds, glycerate 3-phosphate, the first product after carbon dioxide is joined to RUBP, is seen here, and it was the dominant molecule formed after 5 seconds. And also, just a tiny little bit of triose phosphate had been formed after 5 seconds. But after 30 seconds, much more glycerate 3-phosphate and triose phosphate had been formed, as well many other organic molecules. You can see these amino acids here had been formed, as well as sucrose, a disaccharide. Now, in previous slides, I've provided links to short videos on YouTube that talk about Calvin's experiment. Please review both of these videos. And now, on to a last concept, a concept that focuses on Rubisco, the enzyme that fixes carbon dioxide to RUBP. And so the last concept, it's a very interesting biological story that links Rubisco, the enzyme that joins carbon dioxide to RUBP, and transpiration and plant adaptations to dry environments. This is a cross-section of a leaf. We know that these cells are densely packed and full of chloroplasts and function very heavily in photosynthesis. We know that CO2 enters the leaf through stomata. Now, open stomata allow carbon dioxide into the leaf so that the CO2 can diffuse into these cells in the palisade mesophyll, and Rubisco, the carboxylating enzyme, can then join the carbon dioxide to RUBP. However, open stomata also allow water to escape the plant. This could be a problem in dry environments. Here is a micrograph displaying a stoma. Stomata, the plural, are critical in allowing carbon dioxide to enter the plant, but they also allow water to escape. This is potentially a problem in dry environments. You may be asking, where does Rubisco come in on this? Stay tuned, it's coming. So here it is. There's a flaw with the enzyme RUBP carboxylase, otherwise known as Rubisco. The flaw is that Rubisco has affinity for CO2 and oxygen. This is a problem. When Rubisco fixes oxygen instead of carbon dioxide is not being fixed. Thus, the photosynthetic rate is reduced. Now, if a plant kept its stomata wide open, this would provide high concentrations of carbon dioxide to Rubisco, and this would reduce the problem of having oxygen potentially outcompete carbon dioxide for Rubisco. But if stomata remain wide open, water loss is increased. Now, this is not a problem for plants that live in wet environments, but it is certainly an issue in dry environments. Rubisco's affinity for oxygen gets worse. Rubisco, when it fixes carbon dioxide, is called RUBP carboxylase. When it fixes oxygen, it's called RUBP oxygenase. But when Rubisco fixes oxygen instead of carbon dioxide is actually released, lost from the plant. This problem, the problem of Rubisco's affinity for oxygen and carbon dioxide, with carbon dioxide loss if oxygen is fixed, is called photorespiration. Because when Rubisco fixes oxygen instead of CO2, it appears that the plant is respiring, releasing CO2, while it's photosynthesizing. One adaptation to deal with the problem of Rubisco's photorespiration, its affinity for oxygen and carbon dioxide, is seen in plants living in dry environments, and it's known as the C4 anatomy. Carbon dioxide is fixed by an enzyme called PEP carboxylase. PEP carboxylase is not shown in this image, but carbon dioxide is joined to PEP, phosphoenol pyruvate, by PEP carboxylase. Now, PEP carboxylase has a higher affinity for CO2 than Rubisco does. So once PEP carboxylase fixes carbon dioxide, there are various intermediate compounds that are moved deep within the leaf. The details are complicated, as you can see here, but in the end, PEP carboxylase's higher affinity for CO2 allows C4 plants, the C4 anatomy, which is an adaptation to dry environments, it allows these plants to partially close their stomata. The stomata can partially close because of PEP carboxylase's higher affinity for carbon dioxide. The partially closed stomata serve to conserve water. Again, this is an adaptation in dry environments. Once again, you can see PEP carboxylase fixing carbon dioxide to PEP, phosphoenolpyruvate, to form intermediate compounds that are moved to locations deep within the leaf. Ultimately, the carbon dioxide is released and joined to Rubisco, but this is deep within the leaf, so concentration of carbon dioxide is high here, so Rubisco, used in the Calvin cycle, does not suffer from photorespiration, where oxygen is competing with carbon dioxide for the enzyme. The high affinity that carboxylase has for carbon dioxide at the leaf surface allows stomata to close partially, serving to conserve water. So far, I've outlined two photosynthetic pathways. The C3 pathway uses Rubisco to fix carbon dioxide, but due to the need to keep open stomata, C3 plants are well adapted to wet environments. C4 plants use PEP carboxylase with its higher affinity for carbon dioxide, allowing for partially closed stomata. This reduces water loss, and these plants are well adapted to dry environments. And then there is the CAM physiology. CAM physiology, crassulacean acid metabolism physiology, is an adaptation for water conservation in very dry climates. The stomata are closed during the day, which reduces transpiration. Stomata are closed during the day? Yes, but stomata are open at night. CO2 is fixed at night and stored in a compound called malic acid or malate. Then, the next day, stomata are closed. The CO2 fixed into malic acid the night before is then released from the malic acid and transformed into TP using the energy, ATP, and reducing power, NADPH, from the light-dependent reactions that happen during the day. When stomata are closed during the day, this reduces water loss, and this is hugely adaptive in dry climates. In this table, the photosynthetic rates and transpiration rates are given for representative species of C3, C4, and CAM plants. First, notice the quantity of transpirational water loss in C3 plants. Stomata are wide open to compensate for Rubisco's inability to distinguish carbon dioxide and oxygen. C3 plants do best in wet environments where transpirational water loss is not a problem. CAM plants, with their stomata closed during the day, have the lowest water loss. This is perfect in dry environments. Nevertheless, in dry environments, the lack of water limits photosynthetic rates. Now, C4 plants with PEP carboxylase fixing carbon dioxide, the high affinity of PEP carboxylase for carbon dioxide, allows stomata to partially close. Thus, water loss is not as high as it is in C3 plants. And without the problem of photorespiration, photosynthetic rates are higher than they are for C3 plants. This is all very interesting stuff. And that brings us to the end of IB Bio Photosynthesis Part 4.